We left off in the last video with a discussion of some of the parts of a nephron and how it's structured. We're going to finish up our discussion of the urinary system by talking about the formation of urine, the thing that it makes, right? And that happens in 3 steps. It's going to start with glomerular filtration. And glomerular filtration happens here between, where's my cursor? There we are, between the glomerulus, that blob of capillaries, and the glomerular capsule. And it's here where a lot of those nitrogenous waste products, those excess ions and salts and things are removed from the bloodstream. They're filtered out. And then they enter the glomerular capsule. At the same time that that's happening, we're also losing water. So water is also going to be added to the glomerular capsule. And it's that combination of water and all of these solutes, those ions and nutrients and things that are lost through that filtration process that we now are going to call the filtrate. So we're creating the filtrate here through that process of filtration. The next 2 steps, we call them, of urine production, it gets a tiny bit confusing because they don't always happen like step 1, step 2, step 3. They don't happen in sequential order. Glomerular filtration has to happen here in the formation of that initial filtrate, the water and all of those dissolved solutes. But reabsorption, tubular reabsorption, and tubular secretion, those next 2 steps can actually happen at any point along the filtrate's journey through the rest of the tubule. So you can think of those as things that occur, but not necessarily step 1, step 2, step 3. So now we have our filtrate that has accumulated into the tubule, and it's going to start to descend through that loop of the nephron. And it's going to first go down the ascending loop of the nephron, and then first go down the descending loop of the nephron, and then up the ascending loop of the nephron. And the 2 portions of the loop are the cells that make up those portions are very different. So the descending limb is going to be chock full of aquaporins, which hopefully we remember are those protein carriers that are embedded in the plasma membrane that allows water to move, so allows for the process of osmosis to happen. On the ascending loop of the nephron, these cells are going to have very few aquaporins, so very little water is going to be able to be removed from the filtrate at that point. But what it can specialize in moving out is salt. So lots of water can be moved out through the descending loop, and salt can be moved out through the ascending loop. This is the nephron's balance to try to conserve water. So what we're going to see happen is that the body is going to alter the salt content on the outside of the nephron in an effort to force the water that's on the inside to flood out into the outside section and basically prevent our bodies from peeing it out. We're trying not to lose water. This is called a mechanism of water conservation. So we're trying not to lose all of our water through this process of urine. We're going to concentrate the urine, and we're going to do that by using active transport, so a little bit of energy. We're going to use active transport to pump salt out into this extracellular space around the loop of the nephron. And as these cells are pumping salt out and creating a higher concentration of salt on the outside, these cells over here that are loaded with aquaporins are going to experience a lot of osmosis, and they're going to start losing a lot of that water. And the result of that is going to be that we are going to end up with a concentrated filtrate or a concentrated urine. So we're losing a lot of the water out of the filtrate, but maintaining a lot of those dissolved ions, nitrogenous waste products, and solutes. And we do that through the process of reabsorption and secretion. So reabsorption is when something is lost from the filtrate. So all of those examples, the water is going to be lost from the filtrate back into the bloodstream. Salt is pumped or lost from the filtrate back into the bloodstream surrounding the nephron. Those are both examples of reabsorption. We're taking them back into the body. Tubular secretion, on the other hand, is when things are going to be added to the filtrate. So lost from the bloodstream and added back into the filtrate. And those can include really any waste products. So that's going to be more nitrogenous waste products like ammonia and urea, dissolved hydrogen ions, potassium ions, sodium ions, that kind of thing. Anything that didn't get initially filtered from that process of glomerular filtration. So reabsorption is when our bloodstream is taking it back from the filtrate inside the tubule, inside the nephron. And tubular secretion is when it's being added to the filtrate or added into the loop during this process of urine formation as the filtrate is moving through the nephron. And what we end up with, because of that process of water conservation, what we end up with is a very concentrated, obviously it's still a liquid, so we're not going to be losing all of the water or gaining all of the water back, but we are not going to be losing as much water as we would otherwise. I think that we've probably gathered that water is a fairly important molecule for our bodies, as we're made up of mostly water and it fuels a lot of our main functions. So we want to make sure that we're keeping that balance, that water balance within the body. I have a video also that I will be posting as like a supplemental resource. So if you're a more visual learner and you kind of want to see those elements moving in both directions, then that video will help you as well. And you can find that listed in our week 4 module. For your lab for this week in your body systems worksheet for this section, you're going to be looking at the process of dialysis. And hopefully we're kind of aware that dialysis, you would undergo dialysis if your kidneys were not functioning properly. So as you're investigating the process of dialysis, what I want you to keep in mind is that this is simulating the functioning of the nephron and the functioning of our kidneys. So what the lab is going to ask you to do is to label all of these different parts of the kidney, of the nephron, basically from the dialyzer, the dialysis machine. So just to kind of get you started, if you're looking at the nephron, blood enters the nephron from a certain point. So blood is being removed from the patient here. Blood is being removed from the body and added to the nephron here. So that's kind of what that's simulating. So this would be like the renal artery, and then leading up to the nephron. So just to kind of get you started on what that portion of your lab is really asking. But again, if you have any questions as you're working on it, feel free to reach out.